So, well, basically just the main components of the talk will be on what do we actually have in terms of these biotic interactions below ground. And then I jump into this plant soil feedback concept. Probably several of you heard of it before. You for sure, like from other courses, but maybe some others not, but that's okay. I will introduce it, looking mostly at root symbionts, but also the saprotrophic soil biota. Because indeed, as Luc was already saying, in these systems, there's a lot going on at the same time, and they may interact or not, but you would need to consider all of them really if you want to understand the system. And then we have a look at, could there actually be trade-offs in terms of associations with so-called mutualistic beneficial organisms versus the more saprotrophic kind of organisms? Let's see. This is the only kind of a tsunami slide you could say. I also had a lot of fun in making this drawing. I made it a long time ago in PowerPoint, so exploring a bit what can you draw with PowerPoint. But anyway, it is summarizing a bit like from a plant perspective, especially in a natural system where a lot could be going on. There are associations above ground, pollinators, herbivores, parasitoids, what have you, but also below ground. Luc already was talking a bit about some of the root feeders, like some root feeding nematodes, and there indeed plant characteristics related to plant defenses can be very relevant. But of course, also these top-down predators you also have below ground. In the talk of this morning, for me, I will mostly focus on the right side below ground, and that's more what has to do with nutrient acquisition and nutrient availability. So on the one hand, we have mutualistic symbionts that could help with that. I'm sure the legumes have been mentioned before. And what's the main reason for wanting legumes in your system? Or one of the important reasons in terms of nutrient acquisition? Nitrogen. Indeed, that nitrogen fixation. But not all plant species have that. Indeed, certain plant families have that. On the other hand, we also have these associations with mycorrhizal fungi, and actually more plant species have those. They don't really acquire additional nutrients, let's say, from the atmosphere. No, they really scavenge it from the soil. So it first needs to become available to be able to help that taking up. That's why we also have this whole other group of organisms that eat on that organic material and that help to mineralize nutrients to make them accessible again. Okay, the concept of plant soil feedback in a simple way is said, and I think we don't need to make it complex. Basically, the idea is that the plants growing in a living soil will change what is happening in that soil. So the organisms living in and around the roots will respond. There can be herbivores, pathogens, but also the beneficials. And I would say the majority of life in the soil is beneficial, unless you push them in a certain direction. And you have this whole group of decomposers, so the ones living on that organic matter. That's what's happening with these living plants. But as the plant dies, the remains also stay in the soil, the roots, or even some leaf litter, depending on what you return. And as a consequence of these changes in the soil, actually, you can have that the next plant growing there will do better. That's why I have a green arrow, bigger box, or they do worse, reduce plant growth compared to the initial situation, or compared to a fallow, for example. So this whole concept, this plant soil feedback concept, can help us to understand plant productivity over time, and also in, like, when we look at plant species mixtures in a grassland, for example, it can also help us to explain why so many plant species can coexist over long periods of time, because none of them is really outcompeting the others. They're kept in check, because of the feedback type of mechanisms. But of course, ideally, we would like to know how can we predict these effects, and also, especially relevant for this course, is this kind of plant soil feedback effect different for a mix of plant species compared to a monoculture or an individual plant species, but actually mostly comparing now a monoculture versus a mixture of plant species? Will they change the soil differently, leading to a different outcome for the next crop or the next crop mixture even? This is kind of a concept, I think, mostly developed more in traditional ecology, plant ecology, grassland type of studies, but we do know that we can learn from this also in agricultural systems. And on the other hand, there's a lot of experience in agricultural systems that we can also learn from, because certain practices have been done for a long time, like rotations. Basically, that is related to these plant-soil feedback kind of impacts. That's why I put up this review paper, maybe relevant for your group work, I don't know, just to indicate a bit where these concepts actually connect, and also with the differences and commonalities in terms of these type of systems. So on the, well, Lambert was already saying most of these arable systems are really fast, a lot of disturbance, but also a lot of resources, while more natural systems tend to be slower. But then again, you can learn from both for developing more sustainable agro-ecosystems and just also for general understanding. Good. On these mutualistic root-associated organisms, thinking, of course, about mycorrhizal fungi, this is a picture, and actually we even have a video. In this case, we look at ectomycorrhiza. Is it running or is it not running? No. Well, it's also okay not to run the whole video, because basically here we look at the root of pine, and it's just a few millimeter wide, but actually even with your naked eye you would be able to see this. So basically it are like root tips.
You the picture?
You want to do that, Gerlinde? Go.
Well, okay, briefly, just to see whether it runs. But it's available online, so you can watch it. The reason why I still show some ectomycorrhiza as well is because there was some agroforestry, and some people do work with woody plant species.
It's coming up.
Yeah, okay. Just briefly. Otherwise,
watch out for the.
Let's see. Okay. Yeah, and we don't need to play it fully. Maybe you can go here, for example.
Ah, you know it by heart, huh?
Yeah, well, I mean, this is really good material we got here from the Bellecom forest. So here we have some pine seedlings, so you are aware of the plant species. We will get a zoom in. Okay, this is a non-colonized root. These are colonized roots. So you can see already, well, there's a difference in growth rates, but it's the same plant individual. So non-colonized, colonized, they're really densely colonized by these hyphae. And these hyphae are in between root cells in terms of for ectomycorrhiza. At the same time, they also extend in that soil, and that's where these hyphae go and explore nutrients that they take up and trade with the plant for carbon. So the fungus gets the carbon, and the plant gets nutrients, nitrogen, phosphorus. And really, you could see this with your bare eye if you have host plants. You're going to the forest. It's a bit like a glove. It can even provide some protection as well against some herbivory or toxic elements. Yeah, and there's just quite a bit of life going on. Let's see.
You want to stop this one?
Yeah, you can switch screens again. So ectomycorrhiza, yes, you can see it, and actually many ectomycorrhiza also produce fruiting bodies. Quite a number of edible species of fungi are ectomycorrhizal, like your boletus, some truffles, to name a few. But then we come to the, let's see, yes, arbuscular mycorrhizal fungi. That's a bit of a different story. Here we look at the root. This one also works, I guess it does. This is the root, and here we have these thin hyphae. And that's important. That's again this extension into the soil, but the typical structures are really to be found inside the roots. This is the central cylinder. These are cortex cells. So it's happening in the cortex cells, but you only see this fungus after staining the roots. You really need to do some microscopy there. And they don't produce fruiting bodies above ground, so everything is happening below ground. And yeah, this is the most typical structure, the arbuscle, where the exchange is happening in terms of carbon from the plant to the fungus, and nutrients scavenged from the soil, from the fungus to the plant. And these are really inside cells of roots. And now when we look at arable systems, actually this one is the type of mycorrhizal fungi that you would find there, at least when you are dealing with host plants. Quite many arable crops are host plants for arbuscular mycorrhizal fungi, but indeed certain ones like the brassicas, they are not. So yeah, you wouldn't, if you would stay in the roots of brassica, you would typically not see this while companion species that are host would show the colonization. Okay, just so you're aware of this difference. And like in intercropping, if you would have certain woody plants and non-woody ones, you may be able to use the different types of mycorrhiza. But the movie I just showed you from the ectomycorrhiza, that took 4 months to have just 2 minutes. So they develop slow, but that's really for long-lasting, long-lived plants that have the investment to stay put for a long time. And then it's worthwhile. These are a bit faster, but still it will take typically 6 weeks to get good colonization if you start from spores. Okay, so yeah, the diversification of arable systems or agricultural systems, why would you do that? Indeed, also like above ground, you would try and reduce the pest and disease pressure, but also below ground, and ideally also enhance the beneficial ones. Because get rid of your enemies, but make sure you have enough friends there. You can benefit from both sides then. And in a cropping system, where can you diversify? Well, the cover crops were already mentioned, but of course also main crops. Typically, you would not grow the same crop year after year after year on the same place. You would rotate, that's already some diversity. And these cover crops, at least in our type of climate, where we don't have 2 main cropping seasons, that's a nice thing to do, to still grow cover crops, to keep your soil covered. And they help also with nutrient retention and all sorts of things they could do. And typically they're then returned into the soil before the next main crop is sown again. So that creates opportunities to diversify the system. And this is actually part of a rotation that we used in a longer term experiment where also Lammert and Wopke were involved. And just to illustrate the sequence a bit. I will show you results of another experiment that happened before that, but it uses some similar principles. And that's this one. You got this paper, I think, because it was suggested as literature. Okay, you know already something about it, so I'll zoom into it. So basically, we thought cover crops could help us diversify the system, and if we use mixtures, maybe that will yield more benefits than using just monoculture cover crops. So the idea is we could get some pest and disease suppression. We would also like to stimulate beneficial organisms. There could be, of course, some benefits maybe for weeds. They looked at that, we didn't. On the longer run, we would also like to build up soil organic matter. And an important part is also for farmers, I think, to counteract nutrient leaching in between main crops, but also in the end, there should be release again to the main crop. So in this lecture, I think I have the time. The first part will be about this part, and the second part about this part in the same experiment. So we can then see how do they balance out or not. Okay, and as I said, we were thinking that using mixtures may have an additional benefit compared to the monocultures. So how did the experiment look like? And this was work mostly done by a PhD student at the time, Jana Barel, but she's now already in a tenure track in Utrecht. But here we have an aerial overview of our field site that was close to where the strip crops are. Basically, we are here, these little dots. So it's a drone type of image. And these are plots of 3 by 3 meter. You see already the 2 main crops. The pale green, what would the pale green be? It's or endive or oat. And any endive growers in the room? Any oat growers in the room? Yeah, we would need to zoom in a little bit more. Maybe actually here you would see the individual plants still. That's an endive, that's paler green. The darker green is the oat. So this is how the endive look like. This is the oat. So we had actually first a general cover crop, then we had a year of oat and endive scattered across the field. Then the main treatment was really our different cover crops. So we had 4 monocultures, a grass, just English rye, 2 types of legumes, the white clover and the veg, and then 1 radish, so one brassica, the radish, the raphanus. And this one is indeed non-mycorrhizal. These others are mycorrhizal. And of course, the legumes can also fix nitrogen. And then we made mixtures of the lowland perennial and the trifolium, so grass clover and veg radish. What we also had, very important, was fallow, the fallow control. So no cover crops in between the main crops. Originally, we didn't have that in our plan. But I had a meeting with farmers and they said we need to have a fallow. Otherwise, why would we do anything on our field if it's not really having any benefits? Correct, totally correct. But we were confined with 120 plots in our fields. That's what we had booked at Uniform. So we had to squeeze in the fallow, which we did. But that meant that actually all mixtures of cover crops were seeded in half a plot, but still replicated over the 5 blocks. So the radish vetch or the English rye clover were in half plots of 1.5 by 3 meters. But okay, so we still fit in all the treatments, and luckily we had the control. And then still we had some mineral fertilizer in that field happening as well. And then in the second year in June, mid-June, we looked at mycorrhizal colonization in the main crops. In terms of mycorrhiza was both microscopy and also sequencing work. So with microscopy, you can really see this colonization and which kind of structures. With the sequencing, you can really tell which species of mycorrhiza are in there. And we didn't only look at mycorrhiza, but also at some other fungi in those roots. And then, oh yeah, voila, that's a drone image. Actually, the nice thing is that it wasn't only the image, but we managed to collaborate with the remote sensing group, also with 2 master thesis students that also resulted in 2 papers, because many of the important characteristics you can indeed derive from the air, linking that to the legacy of these different cover crops. And then we had some parameters, of course, of the main crop and of the cover crops as well. Good. So what happened? First of all, it looked pretty nice, I think. Okay, there was some hand reading happening, but quite many people were happy to help us out because we could share the harvest. This is a bit how it looked like. Here we already see the 2 half plots. That's a monoplot, half plots. Here it's superficially tilled. That was February. Returning the cover crops to the field before the planting of the next crop. And you can see the field was quite close to where they make the compost. Maybe you know that area at Nergena. Yeah, so that worked out quite well. Okay, the second year again, but fully crossed. All these treatments were fully crossed. So fully factorial type of design. Okay, and then of course you want to know what really happened in those roots. So beware, we look at what is really inside the roots of these endive and oats in relation to what was there before happening in the fields, in the soil. So what was left there? The comparison here is the fallow. If we would say we don't grow any plants, so you would think there's no host present, maybe some weeds, but basically no host for these mycorrhiza. So that's a baseline. If we then look at the level of colonization in the roots, we can see here 2 winners. In our case, we have the ryegrass and ryegrass clover. And I had to say that we had this replacement design, the 50-50 proportion. Sorry, I forgot to say that. So what you would have expected is for ryegrass clover that on average it would be, well, yeah, somewhere here, I guess. Maybe that was kind of an average, but typically we find responses more like ryegrass. And here we distributed the total colonization, so any kind of structure, and the arbuscles. So we found quite a lot of arbuscles as well, indicating we had very active mycorrhizal colonization in these 2 crops.
Is this the average of the colonization in endive and oat?
Yes, correct, because actually for the total abundance in those roots, it didn't matter which main crop. Correct, yeah. So that's why we summarize it like this. Yeah, I can say more about it, but indeed, if we then look into also microscopy based into the other fungi, not mycorrhizal fungi, that's these, we see clearly we have many more non-mycorrhizal fungi in the roots of oats grown after different cover crops compared to the endive. One reason for that could be that actually endive is shorter on the fields, that's planted in May, while the oats were already seeded in March. And then again, we have 2 winners. Again, the oat grown after the ryegrass or after the ryegrass clover yields the highest abundance of non-mycorrhizal fungi. And these non-mycorrhizal fungi, that can be a mix of pathogenic fungi or more saprotrophic ones that still get into the roots. And of course, yeah, part of the reason could be longer time on the field, but also this is the same plant family, the grass and grass might not be ideal. Yeah, so already a potential trade-off, you could say. Even though in the sequencing we didn't find really high abundances of very potent pathogens, but still it doesn't mean that they're not there. Then looking at the sequencing results, so that's before this was just percentage colonization, what you see in the roots. Here, this is the fungal richness, so purely sequencing based, like we mush up the roots, we extract the DNA, specifically DNA of fungi from those roots. And then we see how many fungi do we have in there. On the one hand, the white bars, that's a mycorrhizal fungi. So within one and the same roots, you do get more than one species of mycorrhiza. On average, yeah, well, across all the 10 for the endive and about 15 for the oat. So higher diversity of mycorrhizal fungi in the roots of oat than in the roots of endive. Could be, again, longer period of time, or a combination of, well, some additional species that they could pick up. Yep. And then for the other fungi, well, that's way more diverse than the mycorrhiza. That's like up to, well, 250 different species, or OTUs, operational taxonomic units, in those roots. Slightly less in the endive. So, well, the mycorrhiza are not the most diverse ones in there on this whole fungal community, but of course they can play an important role in the functioning. Then if we look at the diversity in relation to the cover crop legacy, then we find, especially for the amount of mycorrhiza in the roots of endive and oats, that when they grow after radish, we do have a lower diversity of mycorrhiza ending up in the roots of our main crops. So there is some suppressive effect of the radish compared to the fallow in terms of the diversity of mycorrhiza we get in our next main crop. So some suppressive effect there. Not on total colonization, that was similar to the fallow, because radish is not a host plant. And there are some ideas that actually the toxic compounds of radish could even suppress some fungi. But yeah, we only saw it really in the diversity of the mycorrhiza fungi. So yes, things are happening. So the summary of the first part is that, yeah, in these arable crops, this root colonization in the main crops is actually mostly dependent on the legacy of the preceding cover crops, both for mycorrhiza and for other fungi inside these roots. So we get more mycorrhiza or buscular mycorrhizal fungi and also more other types of fungi in the roots of these main crops after ryegrass, whether it was grown in monoculture or mix, no difference. But indeed, that selection effect, the most abundant crop in our mixture, grass, clover, was the grass. On the other hand, less diverse mycorrhiza after the radish, and more diverse after the clover. And again, it was similar for the monoculture or the mix. So mostly selection effects according to what we could see. Okay, that's just the first part of the summary. That was more like this part, the symbionts, the root symbionts, some mutualistic beneficial, we think, the mycorrhiza, but also some other symbionts inside these roots that might not be per se beneficial. But we still have this whole part, this dead organic matter from above ground and below ground, so the shoots and the roots of these cover crops going into the soil. So in our case, we didn't harvest the part to feed cattle or anything. Everything was returned to the soil. And so this recycling of nutrients will become very important as well in this kind of system. Okay, so again, we're there in a typical sequence of main crops and having these cover crops going in, typically like February, March, depending on the time when the main crop would come in. And that's when the feast is happening below ground, all this kind of fresh food. If the temperature is okay, whoops, the microbes start feeding on that. And then on that, there will be some soil fauna feeding on these bacteria and fungi. And that all results in nitrogen mineralization at a certain pace. It depends on a number of factors, as we shall see. But a very important part for using cover crops is also reducing this nutrient leaching and enhancing later on, ideally, nutrient uptake by the next main crop. So timing is everything here. But how do you time that? Okay, let's see. Well, actually, in relation to that, we also had 2 papers published before the one on the mycorrhiza, even though it was all on the same experiment. And yeah, probably several crop system analysis people will recognize this name as well, because he helped out a lot also on the structural equation modeling that will feature in this part of the lecture. Okay, so what did we think of? Again, we still have these monocultures and mixtures of different cover crops, but we thought of how would this then work? How would there be an impact on the yield, the biomass of the next main crop, in this case, the oats. In the paper, we have both oats and endive. And because we had this model in mind, but we also wanted to test this model with numbers. So we had to simplify a bit what would be the main factors. So we thought it's the quantity and the quality of those cover crops, whether it's monoculture or mixture, that's going to be feeding into what's going on in the soil, which would relate to soil organic matter and the mineralization of that soil organic matter. But it could also stimulate plant-feeding nematode abundances, because you do have hosts there compared to a fallow. So yeah, that may be a trade-off. That would then feed into effects happening on the next main crop. But I have to mention here the soil mineral N availability. We based that on what was there at the time just before planting, plus what would be mineralized. And that's something we did in an incubation at a nice temperature. How much would become available during the growing season of that main crop. And then, of course, there could be factors that are not in these boxes that we could not yet include, but are relevant. And you can test it as well. How important are those? Okay, then the results, and I will guide you through this. Maybe it's actually nice to already show the picture. This is a picture of the oats, same genotype, same age. And you can already see it's probably a picture of oats grown in where we have the two half plots. So a legacy of different mixtures of cover crops. And what we have here on the left side is actually the response to the biomass of this oat after a certain cover crop relative to how that oat was doing after the fallow. So if it's positive, actually this cover crop stimulates the growth of that oat. If it's negative, it means that that cover crop suppressed the growth of that main crop relative to when this crop was grown after fallow. Okay, so yeah, we could, that's a bit like this plant soil feedback type of response. So clearly we see after the grass a suppressive effect on the growth of this oat, while with the clover and veg, clearly a stimulation. So you can already imagine something with the nutrient availability, nitrogen is probably at play. If you then look at these mixtures, and that's indeed what you get, lower plants, paler plants after the grass clover mixture, again, selection effects, because if it would be the average, we would sit here, but really it's way more like the grass. While for the veg, yeah, it's positive, a selection effect, but well, maybe we couldn't really call it selection effect because the veg wasn't very abundant, but still radish veg really promotes this feedback to the main crop in terms of, yeah, and we'll see how that comes. So really very striking, visible in the field, visible even from the drone. So height, nitrogen content of that you can quantify. Does it matter to mix? Yes, it does, but it matters what you mix. So these characteristics of these plants, these cover crops, is very important. And we'll get into what is then happening. Now, these are just some numbers, but it's in the paper. Okay, if we then look at this quantity and quality of these cover crops that went into the soil, leading to effects on these main crops, what we see here is on the one hand the shoot biomass, so above ground biomass of the grass, the white clover, grass clover, radish, veg, even in relation to the previous main crop, but okay. So we can see a clear winner, radish, veg in terms of total above ground biomass. But of course, as was already mentioned, I think also by Lammert, don't forget below ground. So here we have the root biomass, and actually there's a lot of root biomass going in via the grass. Maybe not surprising, but still relevant. While there's much less so of root biomass for these legumes of importance. Another thing we see here are these little stripes, and actually these stripes are indicating our expected level based on these monocultures. So indeed averaging what is happening in the monocultures, then we don't see over yielding or so in in the grass clover. But for radish vetch, really combining those groups gives you a higher yield than what you expect significantly. And for the root biomass, it seemed here to be a bit lower than expected just on this, this average, but still. So yes, quantitative differences, clearly. If we then look into the quality and we focus here on nitrogen concentration in that above ground plant material, well, as expected, I guess, we clearly pick out the legumes. Yes, they are double the concentration of nitrogen compared to our other cover crops here. And the mixing does help as well. Like typically, we are indeed in between, even though a bit lower than expected for the grass clover mixture, but especially the radish, which in a mass was really high, it actually in a mix, well, got really enriched with nitrogen as well. So even though the veg wasn't super abundant, it really had knock-on effects. So very nice, okay. Yep, things are happening in the mix, also with the quality. If we then feed that into the model, because we also had this kind of information, I didn't show you the numbers yet, but then this model tested with also some role of microbes, of course. Indeed, that quantity did play a significant role in terms of the availability of mineral nutrients, but also in terms of abundances of plant-feeding nematodes, in both cases positive. So maybe a trade-off, but actually when feeding that back to the biomass, no trickle-down effect there. But of course, we have these unknown parameters that kick in very strongly. So things are happening via the quantity and quality that we didn't capture in our measurements. Any suggestions? What did we miss?
Maybe water retention by
water retention. Yeah, it could be. On the other hand, we hardly ever had to irrigate, so we tried to indeed make that they weren't limited by water. Maybe I can give it away. Actually, this lap incubation didn't really reflect what was happening in the field, we think. So that's also a general suggestion. If you can, try and do things in situ. Take into account, well, also a bit what Luc was saying, like this microclimatic environments, this whole context. So that's what we then try to do, really get out in the field, because we thought also some microbial activities will be relevant there. So in general, thinking about, general ecology is like, can we predict litter decomposition and mineralization? Well, we know climate is very important. So whether you're on the North Pole or in the equator areas, it will be different. But also this litter quality will be very important. And of minor importance would be the decomposer organisms. But of course, if we are within the one and the same field, this climate will be probably not the overruling factor, but this litter quality will be still very important. And we thought probably these decomposer organisms are also still important. How much time do I have? I don't have all that many slides.
Oh, it's okay.
Okay, good. So this was an old idea already, but then we were so interested in these as well. And we thought we have this nice experiment, a field experiment that's easy accessible, just there at Uniform. So let's do the test. We had these different litter qualities. I already showed you some nitrogen data. I will show you the carbon and lignin as well of these different cover crops. This soil was so-called conditioned, we thought, by growing these different cover crops. So we have different situations, and we have a fallow, very nice. What we wanted to know is actually how would that litter decompose, like during this growing period of the oats. And on the one hand, we had all these litters buried in their own soil. So where this cover crop had been grown, that species or that species mix had been grown, and also in the fallow. But of course, ideally, we would want all substrates in all conditions. But we didn't have, well, there was would be too much work, okay? So then we used standard litters, and maybe you heard of this tea bag index. I see some nodding, very good. Okay, green tea, rooibos tea, that's very nice. But then, of course, I was discussing with Jana. I said, well, but we don't grow tea here. Maybe we should have more relevant substrates. So maybe we should do like lignin type bamboo sticks and cellulose paper, that's just carbon. Yes, let's do that as well. So, well, I ended up then doing that because I was so convinced we need to do that. So shaking out all these bags, filling them again, but okay, it was worth it. So in the end, we had next to these also 4 standard substrates in all soils. And we had the great help of a number of master thesis students. This is a master thesis student, was at the time, and she was an intern, digging in all these different litter bag types and digging them out again and then cleaning them, because for some you do get some root ingrowth. The idea is I wouldn't, because the mesh is quite fine, which would enter, let the fungi and bacteria enter, but yeah, some fine roots get in there as well. And that's not part of the litter anymore, right? Okay, yeah, early April, they were all going in, and 63 days later, we got them out again. Things were happening. Let's see if this one works. Just to illustrate, oops, oh no, I'm not illustrating, am I? Okay. Oh, well,
The mouse is on the,
where is the mouse?
it's on the, yeah, move to the left, yeah.
Oh, no, maybe I don't have it.
There was a mouse on the screen.
Yes, but where do I have the link?
But it's an extended screen. Move to the right, move to
I see it here. Ah, cha, to the right, yeah, but then it's not here.
No, that's right.
But it has to be here, right, to play it or not. And it doesn't matter so much because you already see here, well, even you see a root here. Actually, this is a green tea decomposed way faster. Here is the rooibos tea. At some point, we have fungal growing out there, so even a mushroom, and then things eat that. Just to say activity is happening. Okay. Anyway, back to our different cover crops. So here we have these different cover crops again, and different parameters. The nitrogen I already showed you. Here we now have the carbon, and here we have the lignin. So for nitrogen, you know already a difference. Carbon, also clear a difference. Actually, not so much carbon in the radish compared to the veg, for example. The others are more comparable. And then the other one, lignin, not so much in the radish, actually quite a bit in the veg. And the others in between. And the mix was also a bit in between. So if I would then ask you in terms of decomposition rates of substrates, and I know some in the room would know that, what is like a main parameter of litter that people would quantify or would kind of suggest is important to say it's going to decompose faster slow? Do you remember? Well, the litter itself, the litter property. So the organic material,
The CN ratio.
CN ratio, indeed, that's the most common one that people would name, and that's a good start in a way, but it's not the full story, because carbon comes in different forms, nitrogen as well, but in this case, carbon. So if indeed we would look at CN ratio, then the radish would be 20 and the veg would be 12. So you would totally think the veg would decompose faster than the radish. But I can tell you that's not the case, because if you look here then at the decomposition rate constant, on the one hand, when it was buried in fallow, that's in white, or when it was buried in its own soil, we see that actually the fastest decomposing one was a radish, and the slowest decomposing one was a veg. So we do think this lignin actually played a big role. And you can also see this trend that actually in their own soil for the radish and radish veg, they decomposed faster than in the fallow. And as a follow-up, we luckily, nicely, we also had these 4 standard litters that were buried everywhere. So in the fallow as well as in all the different cover crop soils. Also here we have different CN ratios. The green tea, the rooibos tea, green tea would go faster indeed. Then we have the filtered paper and the bamboo. Is there a difference? Well, yes, in fallow, which is decomposing fastest, the highest K value, that was indeed this one, the green tea, lowest CN, okay. But then the next one in line is actually the filtered paper, not the rooibos tea, even though it has a quite high CN ratio. And any idea what makes filtered paper decompose relatively fast? What would you need?
Maybe short carbon change.
Maybe what, sorry?
Short carbon chains.
Well, but they're still polymers, and then microbes act on that. But what do these microbes need to be able to act on that?
Surface area.
Surface area, yeah, but they act, especially when they are with more and more. What do they need to multiply? Water, yeah, but you can't grow on water alone. Yeah, okay.
Dissolved.
Well, they have carbon, more than enough here, but not so much nitrogen. Correct. So they get that carbon, yes, but in order to really grow and multiply, they also need nitrogen. So actually, they scavenge that typically from the soil environment. And that brings us also to then the role of these legacies of these different cover crops in terms of influencing these soil microbial communities. Because if we look then at that filtered paper, yes, it decomposes faster than some of these substrates, but even looking just at the filtered paper, it does decompose a lot faster in the plots with a legacy of radish or radish veg. So double the speed.
But why was the rate of the rooibos tea so low?
Well, so the rooibos tea is not just CN, but really many more complex molecules, so also including lignin. So in the original paper of the tea bag index, they listed all the properties they could measure on those teas. Yeah. But it's still a nice way because they're really standardized initial weights and quite low energy to put them in. So here clearly things are happening, and we also had some data on biomass of the microbes in the soil. So indeed there was a bit higher microbial biomass in the plots after radish veg compared to the fallow. Not hugely, but actually this activity of having that easy food leading to mineralization because of their activities feeds into nutrient availability to decompose also other materials that have carbon but lack the nutrients. So you really get this trigger, kind of a primary, you would call it, for enhanced decomposition and mineralization. Yeah.
Radish. That's not the one that makes sense. Or is the most nitrogen, no?
The radish and radish veg, yeah, I go back briefly. So the radish was very high in biomass, let's say. Not so high, well, wait, the one before, not super high in nitrogen, similar to the grass, but it is low, especially low in lignin. Lignin or complex carbon molecules. Oops, here. So this is, okay, less carbon compared to the veg, but easy carbon compared to the veg carbon. So it's not just carbon, but is it sugar, really easy, or is it chewy stuff that you need to have more enzymes and more? So that's where it comes from. Also, if you see these decomposing, it also becomes quite mushy easily. So yeah, lignin to N in this system is very important. And then looking at the radish veg combination, there you have also a richer in nitrogen and a lot of biomass. So that is really a boost to that whole soil life. You see, flop. But in the end, yeah, it's quite equal here in decomposition rate. But really, it's like doubling the speed. So yes, things are happening, especially in situ. Because actually in this incubation, maybe it was a bit silly, but the protocol for incubation for nitrogen mineralization from a soil is that you do sift the soil first and then put it in bags at a certain humidity, and then you incubate it at 20 degrees for a certain amount of time, and you see how much nitrogen becomes available in that time. But yeah, we removed some of that litter stuff. We should have done both probably, because in the field it's all together, right? Okay. This part of the summary you already saw, then we can add to that, because that was on the mycorrhiza and other fungi inside the roots. Now we also have actually the yields of these main crops. They were highest after radish grown in mixture and monoculture and mixture. So that is not the kind of treatment where you have most mycorrhizal fungi. So mycorrhizal fungi are probably not doing the biggest job in this system in terms of supporting the yield of the main crops. And I say main crops because we had similar responses in the endive, although a bit less high because shorter or later growing season. And these cover crop legacies on decomposition and nitrogen mineralization, I think we can generalize them a bit more than just sticking to these specific species we used. This quantity and quality are super important, and in terms of quality, the CN wasn't saying so much, but really this lignin to nitrogen was more of a key property in our case. And so it's a quality component, but not only the quality, it's also the triggering of that quality-quantity combination on this microbial activity that really makes how then these nutrients will become available while the main crop is growing. And in our case, again, our type of system, there were some pathogens, yes, there were some root feeding nematodes, yes, but they never reached above the threshold that you would really get damage. If that would be the case, then the story would be probably slightly different. And with that, many people to thank, of course. And for now, is there still room for questions?
Of course.
Good.
Okay.
So if something wasn't clear, or now I have some hidden slides, but then I can.
Okay. Thank you very much.